Hi, everybody. I am recording this as a video for you guys to be able to look through and for us to stay on track for our visual system review. So the first part, we're gonna break this up into two components. Basically, we're going to go from the retina and talk about sensory systems and how the retina is basically put together, and then how that information is transferred back to the thalamus, the lateral geniculate nucleus. And then from there, in the next lecture, which will be on Tuesday of next week, we'll go through how it goes from the thalamus back to the cortex and a lot of that complex processing within the cortex itself. So we'll be spending a lot of time talking about the visual system, not as much on somatosensory, and none on hearing. And it's not because hearing is unimportant, but I think the depth that we go into for vision is helpful because it's one of the most carefully studied sensory systems that we have and the most thoroughly studied. And some of the principles that we find there are connected to what we see in hearing and other types of sensory modalities. So it's a useful kind of primer, if you will, for the concepts that are behind some of the other work that people have in the other systems. So let's go ahead and get started here and see if I can do this right. Okay. All right. So the first part of this lecture today, I'm going to go a little bit more quickly, and then there's in the latter part, then we're going to slow it down a lot because some of those concepts get a little trickier. So it's going to feel a little point and shoot at the very beginning, and then we'll get a lot more slower as we move into it. So basically, what's the significance of vision? I don't think that's much of a difficult one to kind of come up with there. But obviously, we need it to be able to help, modulate and be able to navigate a dynamic environment in order to maintain homeostasis as an organism. And so what we want to try to appreciate is basically how our body is able to convert the sensory stimuli of this dynamic environment into electrical signals that our brain can actually be able to process. And so the retina is one that has photoreceptors that helps convert light energy into neural activity and helps detect some differences in the intensity of light. And then the lateral geniculate nucleus serves, that's a thalamic, or in the thalamus, a thalamic nucleus within there, so a cluster of specialized neurons within the thalamus that serves as that first synaptic relay in that primary visual pathway. So we go from the retina to the thalamus, and then what happens is that it goes from the thalamus and then goes to the cortex, and that's where the interpretation and memory is ultimately processed. So that's kind of our path that we're going to be traveling here. And properties of light, we all know this from physics, but light has it's light and electromagnetic radiation. It has wavelength, frequency, and amplitude, and we have different kinds of colors of orange, red, blue, and violet. And the meditation of this is just briefly to help us remind us that we're basically converting this stimulus into something that our bodies can actually be able to process. And so we need to have a type of sensory receptor that's able to discriminate between these different types of wavelengths and amplitudes and respond accordingly. And so what we'll find is that we have receptors that are tuned to picking up on some of those specializations of light. Likewise, we also have the eye itself is structured so that we can actually be able to manage the light input that we get. And so there's issues that we have to think about in terms of reflection and absorption and refraction. And we won't spend as much time on this, but basically optics, if you think about, the field of optics is basically trying to understand how light rays and their interactions are being managed. And we'll talk a little bit about how our eye is able to help manage some of the issues with reflection, and then and focus it so that we're having absorption in the right area of it. Also, the lens will help us. We'll be using refraction to help bend the light rays to the back of our retina. So if we look at the microscopic anatomy of the retina, and this is the receptive field of visual input that we have, it comprises one of the key receptors that we have for light and light energy are what are called photoreceptors. And so these are responsible for helping to convert light energy into neural activity. And so what we have is what they call sort of a direct or vertical pathway. In this case, the flowchart is shown over here on the left, where we're looking at photoreceptors, then feed into what are known as bipolar cells, and then those in turn feed into ganglion cells. So what's interesting about the eye, and you'll see it here, and you'll notice it here in this diagram, is that photoreceptors are located in our diagram back here at the bottom. Our light stimulus, and let me just see if I can switch this over to a laser pointer. Light usually comes in this direction. In fact, if I'm clever about this, let me see if I can actually use a pen. So light comes in this direction. And so what we have to imagine is that these cells that make up the retina are sitting at the back of the eye. And so light comes in and then strikes on the photoreceptors. And the photoreceptors then carry their information back up to the bipolar cell. And then from the bipolar cell, then it proceeds to the ganglion cell. So this is what's known as the direct pathway. What we also, though, have are some other components that help modulate the activity of this direct pathway. And so we have horizontal cells and amacrine cells that help to be able to shape how these stimuli are actually working. And it's actually a lot more interesting than you might think at first blush. So let's look ahead then at the structure of the eye and kind of think about how that light stimulus is being presented. If you remember, we talked about this direct pathway. And what we have over here is just an illustration of it over here in a small version of it. So light comes in, it gets refracted through the lens and focused on what's known as the fovea of the retina. So that's right down in here. And so that's where we have, we're packed full of these photoreceptors that are sensitive to different wavelengths of light. And we'll talk more about this, but it's a highly specialized area that is enriched with these types of photoreceptors. Just to help us be able to appreciate the rest of the anatomy of the eye, we have the sclera, or the white part of the eye, the cornea, which covers the lens, and that lens helps to shape the light so that it's able to focus onto the fovea. In the back here we have what's known as the pigment epithelium, and so this is really important for being able to help absorb light that comes in so that it doesn't reflect and scatter and add confusion in terms of the input that comes in, and even help protect the photoreceptors from getting overexcited. We also have the optic nerve, and this is where the retinal ganglion cells actually feed into, and they feed into the optic nerve and then out. So the path of information is really interesting in the eye, meaning that it's kind of weird and complicated. So light's coming in here, right? Goes back, hits the back of the eye here, gets processed, and that information process moves outward, and then ultimately goes back down into the optic nerve. And so to repeat that, and I kind of wiggle this in here, so it's kind of hard to see, but let's do a zoom in and see how that goes. So light's coming in, flows in, strikes the back, and hits the photoreceptors here. And as we saw in that previous slide, we have photoreceptors that then are able to pass that information back from the photoreceptor to the bipolar cell here to the ganglion cell. And then the ganglion cells then all aggregate and send their information back to and flow back out through the optic nerve. And so that's the path of information that we have and how it's being processed. So we've then been able to introduce some of the major ideas that we have here, the overall structure of the retina, and then some of the specialization that we have. If we look at this picture here, what we can see is just that the fovea being shown here in the middle, this is a retina that has been partially dissected so that you can actually be able to see the different components of it. So it's a flat mount of that retina. And the optic disc, which is right here in this image here, is where all the ganglion cells like that that are throughout the retina are actually feeding into and sending their information out through the optic nerve. And so here you can see this is the where optic disc is for this particular type of schematic, and then this is what it looks like in the eye itself. And so this actually corresponds to our blind spot. So there's a spot in the eye in which if light happens to fall in that area, we won't detect it. And that's because that's where all of the information and where all the the axons of the ganglion cells are actually projecting out. And they're heading out to the lateral geniculate nucleus. So there's a ton of processing that happens in this area. And there's some details about how we want to be able to think about the structure and everything. So let's kind of dig into that and talk about the laminar organization of that retina. So there's an inside-out type of orientation that's present here. And so I'm gonna go through and just kind of talk about some of those different layers and how light's going through. This is illustrating how light shines down and then hits the back of the eye. And what we can see here is that here are our different layers here. And we're starting to see these different layers illustrated here. So we have our pigmented epithelium. Then we have our photoreceptors. And the photoreceptors have two different kinds of layers. One is the outer segments of the photoreceptor, which is out here. This has a lot of the biochemical components in it that are sensitive to light and will respond to light. Here's the cell body of the photoreceptors here, and then here's how it's projecting out to ultimately meet our bipolar cells that reside within the inner nuclear layer here. There's an outer plexiform, this region here is known as the outer nuclear layer. You can just remember it because it's nuclear, like remember nucleus of a cell or where the cell bodies tend to reside. This plexiform layer, or outer plexiform layer, this is where a lot of really cool kind of processing happens that we'll talk about, and helps to be able to integrate how the different bipolar cells are responding to the sensory inputs that come in from the photoreceptors. The inner nuclear layer is just referring to where we have nuclei present for the bipolar cells. And then what we have is the inner plexiform layer here. So this one's where we have the connection that's made between the bipolar cells and the ganglion cells. And then these are shaped by this green guy here, which is known as an amacrine cell, which also can help influence the tuning of and the shaping of how stimuli are being processed. We'll talk mostly about these guys here in terms of how that information is shaped, but we'll integrate it into talking about how these guys work here. So inside out is basically saying that it's trying to say that it's kind of weird. You would think that light would hit, the photoreceptors first in the outer layer, and then as you moved inwards in towards the eye, then you would actually be able to, have the processing, and then it would shoot out through the optic nerve. But that's not the way it's set up, and I don't think there's a just so story for why that's the case. So what is shown in this diagram is I pulled this from another source, but I thought it was useful because it had some more anatomical details in it that were helpful. The thing that's potentially kind of annoying about it is that it does show the photoreceptors at the top rather than at the bottom. So when you're comparing these two, this one's showing the photoreceptors at the bottom, closest to where we have the pigmented epithelium in the back of the eye. That's the typical representation that we have. In this image here, they've kind of flipped it around, and so they're showing the photoreceptors here, and then here's the outer plexiform layer, the internuclear layer where the bipolar cells are, and then the inner plexiform layer where we had the bipolar cells meeting the ganglion cells and ultimately sending their information out. Pretty much what I said before. What I like about this image, though, is that it helps us to be able to see some of the details that we have here. For example, the pigmented epithelium here. And then it helps to label, and it shows a bit more of the heterogeneous nature of the different cell types and the shapes of those nuclei that we have. And that there's a lot of variability in the size and shape of these cells. And we'll talk about how some of these different cells, based on their size and shape, actually process information differently. We have rods in here, for example, and we have cones, R and C. So these are two different shapes that pick up on different aspects of light. But even in terms of the types of ganglion cells that we have that reside here and here, for example, you can see this one's kind of has a longer kind of projection here, and it's kind of spread out. This is shorter, but these types of differences that we see in the ganglion cell reflect some differences in their characteristics overall. But the simplified version here is the easiest to be able to kind of to think about, and so this kind of gives us our grounds us a bit more. So we have rods and we have cones in this that reside within that are photoreceptors here. Cones are the ones that are sensitive to different wavelengths of light, and then rods are ones that are more sensitive to intensity of light. And then these are passed on to bipolar cells that we have that are tend to be specialized in terms of their inputs here. And so, and these are illustrating two different types of bipolar cells that are picking up on the same type of cone here. And we'll talk about some examples of how that works. And then ultimately feeds into these ganglion cells. So this gives a little bit more of like that morphological complexity that we see, and then is another representation of some of those different layers that we just talked about. And so our visual receptors, those rods and cones here, we can see that we have different kinds of rods and cones that are present. So the rods are typically located out in the periphery, outside of the fovea, and then cones are scattered in the periphery, but are highly concentrated in the fovea, and they have different shapes there. And that helps to reflect that the size of the receptive field, what they pick up on, and then also just some other types of sensitivity differences that they have between them. But as a general type of characteristic, what you have in your rods are these free-floating discs that are present here, and what they call a ciliary stalk that helps to connect those two from the outer segment out here and the inner segment that we have here. And we have these discs that are present that help to pick up on the sensory input that comes in from light. And we have a variation of this, what are described as being infolded discs here for the cones. And when we're looking at these types of rods and cones, and what we see inside of them is that we have these membranous discs. They are containing the photopigment within them here and here, and so it helps to, and they're located within this, as I described before, but now we can see it named here. As an outer segment here, and then we have our inner segment here, and then finally our synaptic terminal, where we have our plexiform layer. So the outer segment is describing this characteristic of a photoreceptor. Inner segment is describing an area of the photoreceptor. And then we have obviously the cell body and the synaptic terminals there. So if we look at the differences in the central retina versus the periphery, temporal periphery or nasal periphery are referring to the sides of the retina that are closest to the temporal bone, meaning sort of towards the outside of your eye, towards your left or towards your right, whereas the nasal periphery is a more medial positioned region of the retina, where we have it. And so you can divide the retina into those two basic types of areas. And then we have the central retina, where the fovea is. And you can see that the structure is set up differently. The peripheral retina has a much greater number of rods in it, represented here in blue, and a fewer number of cones. And that this information that we see here is we move from having these different rods here, that send its information up to these bipolar cells, and then these bipolar cells end up converging their information onto a ganglion cell here. So I'm just going to write G for ganglion cell here, B for bipolar cell here, and then this is the photoreceptor itself. And so we're kind of seeing this flow backwards here. So, okay, awesome. That convergence of information here represents in something in sensory physiology, and that is you're going to have a harder time discriminating items from each other if you're converging information here. So light or activation of, let's say, this particular type of rod here could activate this ganglion cell just as easily as this particular one over here. So if you just shown a light, so you only activate this one, then that would make this fire. And if you move that light over and then shine it on this area, it would still activate the same ganglion cell. And so to your body, and that ganglion cell then passes it on to your brain and so on. And so for your brain, basically, you're not going to be able to tell whether the light hit here or whether the light hit here. And so this convergence of information that happens here reflects that you're not able to discriminate between the position of the light on the sensory field of the retina itself. So that's happens in the peripheral retina. In the central retina, it's much more focused. So there's less convergence of information, and so there's a greater fidelity and representation of it. And so if you imagine that this particular chain that's being shown here is repeated, let's say like over here we have a repetition of that. And I'm just going to kind of draw circles to help represent it here. And so we have, another, let's say, photoreceptor, another bipolar cell, and another ganglion cell here. If you shine a light here, and then it goes to this ganglion cell here, that ganglion cell is able to fire. And then you move the light over one cell distance, and then shine the light over here, it would fire and activate a different ganglion cell. That's supposed to be a G. And then that in turn goes back to the brain. And so your brain has the ability to be able to discriminate between whether you're shining the light here or shining the light here. And so you have a much greater power and ability to discriminate between sensory stimuli as you move it across the length of the fovea, or move it across the retina here. So your central retina is the one that has that capacity to be able to make that greater amount of discrimination. And that's because there's a greater one-to-one type of representation of stimulus of a particular type of photoreceptor here, and then ultimately activation of the ganglion cell here. So convergence of information here means less acuity or less discrimination. And then less convergence here means that you're able to have greater acuity and greater representation of this. So this is a little more high def, if you will, high definition, than what we have over here in the periphery. So the other aspect of this that you may have noticed is that we basically, in the central retina, you have a greater ratio of these cones that are present here rather than rods. And so you're cranking up on the number of cones here. These have a higher, they are able to pick up on color and other features much more readily than the rods are able to. This kind of difference in terms of the distribution of rods and cones is interestingly represented over here in this this top graph here. So I'll just kind of switch this over to a little laser pointer. And so the cool part about this is that as you move away, this is the central part of the retina right here, corresponds to this area here. So these two figures actually kind of talk to each other a bit. And so in that central retina, what you have is you have a ton of these photoreceptors jammed into the fovea, and they're cones here. So And then as you move away from the central retina, you have a greater number of rods. And so this is the number of rods that goes up here. And so this is, you can see here, number per millimeter squared. So, so you're obviously increasing the number of rods that we have here. So that's really cool, because what you've done is that you basically have, this image and this figure I think is really interesting, because we're able to kind of represent not only the convergence that happens in sort of the microstructural types of arrangements of the retina, and then how that can result in differences in information processing, but also how you can kind of see how the rods and cone distribution is different over as you move across the retina, from the more temporal periphery over here to the more nasal periphery over here. And then there's the blind spot. So kind of a neat image that you have there. Okay, so if we look ahead then and see what we have in these types of areas and some of the regional structure, some of this is a bit of a repetition, but it helps us to make some connections here. So the fovea actually has a bit of a pit in it to help the light be able to access the cones a little bit more readily. So you can see this little pit that's present here, where we have a shift and a movement of the cells that's split apart from the central part of the area here. And here's where we have a ton of the rods, or the cones rather, being represented, and then more rods out here in the more peripheral retina. And so this allows a greater amount of visual acuity. And then this one-to-one ratio with ganglion cells we talked about before, where we can see, like, here's a particular cone here, and then here's a ganglion cell here, and there's that ratio, 1-to-1, is being represented here, versus what we have here, where we have many photoreceptors feeding into a single ganglion cell. So, and then here's a repetition of our different layers here, talking about the outer nuclear layer, inner nuclear layer, and ganglion cell layer. So another representation showing that cones are most highly, the highest density in the fovea. Just thought this was kind of cool. Here's our blind spot. Here's the cones being shown here, and the highest density here. Rods are relatively sparse in that area compared to the peripheral retina. So how do these guys work? And what is it that they actually, how do they function? And so in the dark, if we look at a photoreceptor and we look at how the biochemical cascades relate to a physiological response of a particular cell, what we're looking at is, here's our membranous discs at the back here, and then we're looking at information flow moving out this direction. And what happens is that we have these membranous discs here that are controlling a nucleotide known as cyclic GMP, and this nucleotide then can help control what are cyclic nucleotide gated ion channels. And so cyclic GMP binds to and opens sodium gated channels that allows sodium to then end up flowing into the cell here. I think the thing to kind of keep in mind as you're looking at these photoreceptors is just that almost everything is backwards. So you have to tread carefully as you're going through and trying to memorize or think about how these particular cells work. It's kind of amazing. So I'll illustrate a couple of them. First of all, when we think of ligand-gated channels, we typically think of ligands coming from outside of the cell and then binding and opening up an ion channel that allows sodium or something else to flow in. But that's not the case with these photoreceptors. The ligand is actually internally created, and it's cyclic GMP, and it binds in the internal side of that ion channel and then allows sodium to come flowing into the cell. The other thing that I kind of like about this depiction here, it shows that, well, this is depolarized, so you get this sort of constant release of exocytosis and neurotransmitter, and that feeds on. But the nice thing is that if you're sort of a visual learner in a way, you can see that this is dark in here. This is what's known as a dark current. So the other thing that's kind of backwards about these receptors and these cells is that not only are we talking about these being able to have that we have an internal ligand that's kind of being generated and causing this ion channel to open up. But this is happening in the dark. And so it's the absence of stimulus here that allows exocytosis to occur chronically. So you're constantly having this dark current here. You can kind of see that represented here. When light is shown onto a photoreceptor, what happens is that then activates a process in which cyclic GMP gets broken down into GMP. The absence of having this cyclic nucleotide opening up this channel here like it has here means that sodium can't drift in as it did before. And so the cell can no longer be depolarized and instead becomes hyperpolarized and no longer releases neurotransmitter. So they describe it as a dark current, another great band name, if you will, I think. But you can see how it's normally at around minus 30 millivolts because of that sort of influx that comes in of sodium. So it keeps it kind of positive relative to our typical minus 70-ish resting membrane potential. And then when light is shown, as is being depicted here in this image here, the cell membrane actually hyperpolarizes. And so that's being depicted in this case. And then it kind of wears off slightly as light goes on, but then ultimately then returns here. And this adaptation is somewhat controlled by calcium and other factors, and we'll talk a little bit about that in a bit. But we can see that this kind of thing is an important concept with the understanding how the mechanism of this unusual sensory receptor works. So this is a close-up here of showing basically a red and a green cone. And what we find basically, and what this is to help illustrate, is that this process that I described in general for photoreceptors, both rods and cones share that kind of dark current kind of characteristic. It becomes specialized in terms of the types of wavelength that the receptors are actually sensitive to. So we have green ones and red ones. And so if we show a green flash onto a green photoreceptor here, one that's sensitive to green light, for example, what will happen is it's hyperpolarized at around like minus 30 or so, and then we show a green flash, then it will go and become. I said this is actually depolarized here, sorry, and then we show a green flash, and then it becomes hyperpolarized, and then we take away that green flash and we actually shine it. In this case, it's red light. In the red light, it's not responsive. So what I like about this figure is that it helps to illustrate that you can shine light onto a green photoreceptor and it won't respond. It'll remain at this sort of dark current / depolarized state. But when you shine the light that it's actually sensitive to, the wavelength that it's sensitive to, that will cause it to become hyperpolarized. And that's where we have this kind of response happening. And then it comes back up again. So we're kind of taking this idea that we saw here of dark current and light current, and instead we're saying for green photoreceptors, this could be red, and it would be the same as being dark. It doesn't pick up on it. But when you shine a green light, then that makes it respond, and you get this hyperpolarization occurring. Okay. So the other thing then that we can introduce, so we've kind of talked about these different types of receptors and all of that, is talking about what's known as this sort of receptive field here. And so what's being described here is an illustration of this of the space or the field of the retina. So just a little bit of the sort of square footage, if you will, of the retina here is being represented. But what's also being depicted is that you're on your z-axis here, which is shown here, is what happens in terms of membrane potential. So if you stimulate with a particular green light, here, nothing, and then, but when you hit this little spot here and you actually shine a green light there, you get this hyperpolarization occurring. And the purpose of this is it's asking this is representing the receptive field of a single photoreceptor, or a single, whatever cell it is that you're measuring. And so to get this kind of an image here, to understand what this means, basically what they've done is that they've stuck some kind of electrode into, let's say, this guy right here. And so they have the electrode here, and they're measuring from there. And they're monitoring it. And then what they're doing is that they're shining light. Let's say they shine light here, nothing. They shine light over here, oops. Darn it. They shine light over here, nothing. They shine light here, as I drew before, nothing. And you had your electrode that's being kind of recording from here, right? So that's all represented by, there's no hyperpolarization being done in here. So we're looking at what's the receptive field of this particular photoreceptor here. And so as we move across and we shine light as it goes across here, we can see that there's no response. Then finally, when we happen to shine light right at the place where it needs to go and hits the photoreceptor square on, that's when we see hyperpolarization occur. And so that's how this is represented. So I hope that makes sense. I know when I was first being introduced to this, nobody explained this. So it took me a long time to figure out what the heck this really meant. So I know it can be a point of confusion, just because, at least it was for me when I was first being introduced to it. But once you get it, then it's really simple. And so maybe, we've just kind of, gotten right to that point where it's really clear. But so this is basically trying to represent, it's saying, okay, we're showing how a single cell here that we're recording from, what is it in its receptive field? When does it actually respond to it? And then what color light does it actually respond to? Is another type of question you can ask. But here it's just asking, when does it actually get stimulated? Okay, so now let's look at phototransduction, some of that biochemistry that's beyond there, and then we'll come back to talking about some of that phys that's part of it. So photoreceptor transduction that we're considering here. Let's look at what happens in rods. We have depolarization that occurs in the dark, that dark current that I was mentioning to you before, and then it becomes hyperpolarized in light. And so there's an opsin that's in rods. It's known as rhodopsin. And there's only one type of opsin there. This is in contrast to what we have for that's in contrast to what we have for cones. And what we are looking at then is that these are activated. This kind of protein is actually changes its conformation in the presence of light. So in the dark, then we have retinal that is relatively inactive. And it's present within a protein here. So this is a disc membrane. So first time I looked at this, I kind of got confused. So I'll make sure you get it. Remember, like these discs here? That's where we have these little opsins all crammed in there. And it's opsins crammed into that disc membrane. And then within that, we have this retinal that's present. And so in the inactive state, which is in its trans state, then we see that we have this kind of kink that's present. Light shows up and then changes the conformation of that chemical, and that in turn helps to trigger a change in the conformation of opsin itself. And so what ends up happening then, if we look at a sort of a larger picture, is that we have the rhodopsin being present here. There's our little presence of our retinal stuck within it. And then the opsin protein itself, as I was describing before. In the inactive state, in the dark, as we can see here, what we have is that cyclic GMP is being constantly produced. There's like a constant sort of production of cyclic GMP. It's binding to and allowing sodium to flow in, all about the dark current. Awesome, great. And then you get the depolarization and so on. Then what happens is that if light comes and hits rhodopsin, as it's being described here, this activates a phosphodiesterase through what's known as a G protein. So this G protein then basically takes on GTP and then it gets activated, and then that in turn then activates this phosphodiesterase. The phosphodiesterase is what's responsible for helping to break down cyclic GMP into GMP. You may have remembered this phosphodiesterase as being one component that I described in learning and memory, and that's for what the modulation of cyclic GMP and that particular type of system. And so, and it was a target for this the company called Tetra that's located up on the 5th floor of the Cook DeVos building at CHS. And so I mentioned that in passing before, but now we're seeing it again. So it's just kind of interesting to see these things kind of echo through these different systems of our body. Nonetheless, phosphodiesterase is the thing we want to focus on, is that this is what's chewing up the cyclic GMP and removing that sort of constant signal that's being produced. Absence of cyclic GMP means that this channel will close, and then the cell will ultimately, and then if this is a class, I'd ask you, does it depolarize or hyperpolarize? So, think about that. And what happens? Does it depolarize or hyperpolarize here? Is the cell depolarized or hyperpolarized here? All right. So this phototransduction that we're talking about in rods is more specialized in cones. It's similar, but we have different types of opsins that are sensitive to red, green, and blue. And so this allows us to be able to have spectral sensitivity to it. And so that's being depicted here in terms of wavelength and these types of opsins that we have within the different cones. And so these cones are sensitive to different or specific wavelengths of light. These opsins help to allow this to occur, and so this is being represented here. The reason I added this was that this image is really similar to what we had on the previous slide. But what's nice about this also is being able to see how these green cones, for example, if you have green light coming in, then you can see how it causes this physiological response, a greater hyperpolarization that's occurring here in the cells that are they're being shown this green light. So just that kind of change and differences, go through and just make sure you know and you can understand why they respond. So green light, you have a preferential activation of these green cones here, and the red cones are really kind of nonplussed by the whole situation, although there is some kind of responsiveness being depicted here. With regular light mixed between green and red, you can see that there's a kind of a responsiveness that happens in both cases. And then with red light, you can see that there's a greater amount of responsiveness here in the red. And so with light energy, there is some response. Like red light can cause a response in some green cells. But obviously there's a kind of specificity to its action. And so this is a bit more of a careful type of description of what's happening than what we may have seen here, where we just said, okay, red light, no response, and then you shine a green light, you get the hyperpolarization. So it's a little more subtle than that, because a lot of these green cones do have some sensitivity to red, but their focus, if you will, or their sensitivity is mostly around the green. So there's adaptation that can occur. Typically, in daytime vision, you're mostly all cones. It takes about 20 to 25 minutes to shift over to this nighttime vision, and requires dilation of pupils and causing a regeneration of this rhodopsin that's present in the cones themselves that gets bleached relatively quickly, and some adjustment of the functional circuitry. And calcium can play a role in this in terms of being able to help indirectly regulate the amount of cyclic GMP that's present. And so by tweaking the amount of cyclic GMP, you can ultimately tweak the amount of depolarization that you get out of the cells. So when we think about the processing that can happen, moving from the biochemistry to thinking about how processing can occur, we end up being able to look at these, we can sort of revisit the structure that we have of these receptive fields. I'm actually going to stop here though, and have the second part be a second video. So we're going to take a pause, and I'll load the other one as a second video to take it from there. So in sort of recognition of the switch.